So the next speaker in the session is Professor Eckhard Mandelkow. Professor Mandelkow has a background in physics, and he did his PhD in Heidelberg and then moved for some time to Brandeis University in Massachusetts in the States. And then he went back to Germany, in particular to Hamburg, where he's been there till 2011, I believe, when he moved to the German Center for Neurodegenerative Diseases in Bonn. Professor Mandelkow, together with his wife Eva, which is also present here, has been leaders in the field of tau protein structure, aggregation, and cellular functions for many, many years. He's been recipient of a number of awards, including the MedLife Award and the Potemkin Award, and among others. Today he's gonna talk about the role of tau protein in Alzheimer disease and other tauopathies, structure and cellular functions. So thank you, Eckhard, for being here, and welcome to Spain.
Thank you very much. First question is, what button do I have to press in order to get this going? Oh, here we go. Thank you very much. We have a slight problem with movies, but I think it's going to work. So, talking about tau in Spain is a little like carrying owls to Athens, right? As the Greek proverb says, there's so much tau work that has been done over the years here in Madrid and in other places, mainly in the school of Jesus Avila, who's sitting in the first row, smiling. Well, thank you very much for the invitation. It's nice to be back. And I remember many other very interesting meetings that took place over the years in different cities of Spain. But my first time in Alicante. Okay. I'll just review a few things that have happened in the lab over the past several years and give you a flavor of what we've been doing. Let me first start. I mean, one of the things that were dear to our heart was the structure of tau, the structure of microtubules. Most tauists somehow have an origin in microtubule work. There were cell biologists working on microtubules. Tau turned out to be a microtubule-associated protein, and after working a while on microtubule-associated protein, people found themselves in the middle of Alzheimer research because in the year, I think, 85 or 86, it was discovered that the main component of the paired helical filaments in Alzheimer's disease was the tau protein. So that then led to a number of issues and I have a few things listed here that I'd like to cover. So let me just go through these topics and let's see how we go on. So first, the physiological and pathological functions of tau. As you all know, tau is let's see, yeah, okay, so is one of the deposits in Alzheimer's disease, occurs in neurofibrillary tangles within neurons, in contrast to the amyloid plaques that are outside neurons, and if you isolate it, you get these filaments. They're called paired helical filaments, although they're not really paired helical, but they look like it. And they have a characteristic periodicity of 80 nanometers, which distinguishes them from other types of filaments. Now, one of the important things about tau is the spreading, which is throughout the brain, which was discovered early on by Braak and Eva and Heiko Braak, actually. So these are now called the Braak stages, and you see that early stage is just here in the entorhinal region, then covering the temporal lobe, and then eventually covering the entire cortex. And this correlates very well with the clinical symptoms, and that's why you know, we can correlate the deposits that we see in the brain very accurately with the clinical diagnosis. Now, I mentioned that tau is a microtubule-associated protein, so a quick word about microtubules. Microtubules occur in, does this move autonomously or not? I think it does. Let me see.
It's moving.
It's moving, okay. So we have here, a neuron with a long extension, that's the axon. That poses a transport problem for the neuron because things at the synapse have to be fed from the cell body. The transport system of the axon is, of course, the microtubule system. Microtubules can be thought of as tracks, and which have motor proteins running along them in the forward direction kinesin and the reverse direction dynein. And like a railroad track, it has to be stabilized by ties, and the ties for the microtubule system is the tau protein and other microtubulous associated protein. They're shown as black hooks here. So the hand-waving argument about the function of tau is that it stabilizes microtubules. It does actually a lot more, but that's the most basic function that was discovered early on by Mark Kirschner and colleagues back about a little more than 50 years ago, I think by now, or 40 years actually. So one of them is dynamic instability of microtubules, which means microtubules are not simply static, but they grow and shrink, and that enables them to adjust to shape changes within the cell. And also the other thing is, of course, the axonal transport is shown down here. Now, there are also a number of hypotheses of how tau can be toxic to cells. I mean, the honest answer is we're not really sure, because tau, as I'll show you in a minute, has many different proteins that it can interact with. But the microtubule-centric point of view is that, first of all, in a normal microtubule, motor proteins can move up and down the microtubule, and the microtubule is only sparsely decorated with tau protein, and therefore a motor protein which is driven by the power of ATP hydrolysis has no problem of moving along and pushing the tau out of the way if necessary. However, two scenarios can happen. One is that overdecoration of microtubules, local overdecoration of microtubules, can inhibit the access of motor proteins to the motor protein, and then in that case you have a retardation of axonal transport. That's one of the scenarios, and which can then of course has effects on the nourishment of the synapse. The contrary view is that if the tau does not bind properly to the microtubule, for example, because it gets phosphorylated by certain kinases, then two things happen. The tau can come off the microtubule, and when it comes off the microtubule, it is then free to associate with itself for reasons we don't really understand. But anyway, in the course of that, it can form oligomers and peritelial filaments or tangles. And the other thing that happens is that the microtubules can fall apart, and therefore the tracks for transport disappear. So you have an obstacle in the brain that can inhibit axonal transport, and the tracks themselves are inhibited. Now, I should say, a cautionary tale, this is all pictorial work. I mean, the disappearance of microtubules is observed in Alzheimer's disease, and of course the tangles are observed in Alzheimer's disease, but whether they're actually causal or the only causes of the pathology that we see, that's a different issue. So what protein are we talking of? Tau is interesting from several points of view, and the most interesting thing from a biophysical point of view is that it is a totally non-structured protein. So most proteins, as you know from the textbook, have a nice structure in terms of alpha structure, beta structure, etc., hydrophobic inside, hydrophilic outside. Tau is none of that. Tau is very hydrophilic, as already discovered early on, and therefore it has absolutely no reason to fold. And as a result of that, it does not fold in the globular shape. So it is accessible to many other proteins that occur in the cell. For example, it can be modified by many different enzymes, kinases, proteases, phosphatases, etc. And this makes the work on tau a little bit complicated. But anyway, here is a picture of the domain structure of tau, and the thing that I want to point out is this box here is the so-called repeat domain, 3 or 4 repeats. The green ones are alternatively spliced segments of tau. Anyway, the repeat domain is the one that interacts with the microtubules in a physiological sense, but it is also responsible for the formation of the peritelial filaments, and therefore the physiological function and the pathological function are encoded into the same part of the molecule. So it's like a switch, either pathological or physiological switch. The other thing shown here is the many phosphorylation sites that tau has, many of them threonine proline or serine proline motifs that are suggestive of proline-directed kinases, and many of us have worked and analyzed these kinases and the phosphorylation sites. Let me just say there are actually too many too many and too heterogeneous to really understand, but of course the hope was to find particular kinases that are responsible for the pathological transition of tau, again, in spite of a lot of work over the many decades, it is still a little bit enigmatic what the role of phosphorylation really is. Then the other thing that was discovered about 15 years ago is the mutations that occur in tau protein, which cause frontotemporal dementia primarily. These mutations were called FTDB17 because they're related. So the mutations have two functions. One is they actually usually increase the ability of tau to aggregate, and they decrease the interaction with microtubules, which fits with the general picture that aggregation is bad for the cell, and loss of stability of microtubules is also bad for the cell. Many of these mutations have been used to make cell models and animal models of tauopathy. They work quite well. They cause aggregation in the transgenic mice, for example. I show some examples later. And many people have, for example, looked at the mutation P301L, which or P301S. In our lab, we have focused on the mutation delta K280, which is also located in or near the repeat domain, and therefore would explain why it affects the aggregation of tau. And the other mutation we have looked at more recently is the A152T mutation, which is different from most of the other mutations because A152T is a risk factor for PSP, basically, or FTD-related diseases, brain diseases, but it is located far outside the repeat domain and therefore not immediately related to the assembly. And I'll show you that, in fact, these two have different kinds of toxicity that you can observe in mouse models. Now, what about tau structure? Here, a picture of a microtubule protofilament, alpha and beta subunits lined up nicely and nicely folded with actually alpha helical structure and beta strands in between. So prototypic globular proteins that self-assemble into microtubules, and you see a motor protein kinesin. All of this is solved either by X-ray diffraction or electron crystallography. Also nicely folded with alpha and beta structural elements, and in contrast, a fantasy model of tau sitting in an unfolded way on top of the microtubule. And this unfolded structure shows you that it is an open, loose cushion sitting basically on the surface of a microtubule, able to interact or maybe able to shield the microtubule depending on whose axis has to be controlled. So this is the problem that we're dealing with. And it also illustrates that because of this highly soluble, open, hydrophilic nature of tau, it does not aggregate. And one of the points that I'd like to make is that tau by itself, and I'll show you some evidence in a minute, tau by itself does not aggregate. In spite of the fact that we see aggregates of tau in various brain diseases in a very stereotypical fashion, tau is so highly soluble, it has no reason to aggregate unless we assume that there is some other factor that helps the aggregation. And in fact, I think the group of Jesus Avila was actually one of the first ones, the first one probably to show that things like heparin sulfate or other negatively charged macromolecules may play an important event in helping tau to get on its way to aggregation. So tau by itself does not do any harm as far as aggregation is concerned, but in the contact with other macromolecules, it may do so. So here is just a pictorial representation of how a kinesin molecule would like to attach to a microtubule surface, but it can do so only if it has a free surface, and if there are too many tau molecules like the red one here, then it takes longer to attach, and therefore axonal transport is retarded. So here we did some solubility studies, just to back up what I just said a minute ago. So a lot of people, including ourselves, were trying to crystallize tau. That was, of course, a hopeless task because only globular proteins, roughly speaking, crystallize well and not disordered proteins like tau. But the solubility in micromolar is shown. So in order to do crystallography studies, you look at the solubility. And I show you here the solubility limit for tau full length is about 1,000 micromolar. Repeat domain is about 4,000 micromolar. If you express it in E. coli, you get it up to about 200 micromolar. And if you get it in SF9 cells, it's also about 200 micromolar. In E. coli, it gets expressed without phosphorylation. In SF9 cells, it is expressed in very high concentrations, in very high phosphorylation. But in both cases, they're totally soluble. The tau protein is totally soluble, proving, for one thing, that the high phosphorylation does not necessarily drive tau into aggregation, which is normally the expression you get from this highly phosphorylated Alzheimer tau. But here is the concentration of tau in neurons is about 1 micromolar. So if you have a 1 micromolar protein, and it's soluble to up to 1,000 micromolar, what do you expect in terms of aggregation? You expect nothing, basically, and that's what happens in cells when you express them. The tau, as such, does not aggregate, and that's why it has taken such a long time, actually, from the discovery, let's say, in 1986, as part of peritelial filaments, until the time it was shown to aggregate into in vitro, and that has to do with this very high solubility. Now, let's also compare, for example, here, tau in the interstitial fluid measured by David Holtzman's group, 0.001 micromolar, meaning a nanomolar, and in the CSF it's a lot of zeros, picomolar. So very, very low concentration. All this tau is not really aggregated unless it is complexed with somebody else. So self-assembly of tau by itself is highly improbable. Nevertheless, tauopathy is linked to the aggregation of tau, in most cases anyway, with a few exceptions. And therefore, we have to, if we want to understand the process of this pathological aggregation, we have to focus on aggregation-inducing factors. And the other conclusion is phosphorylation is not likely to be responsible for aggregation by itself. Certain phosphorylation sites may play a role, but we don't know really which ones they are. So here, another picture of the structure of tau derived from NMR studies that were done with our colleagues at the Max Planck Institute in Göttingen, Markus Zweckstetter and Christian Griesinger, showing basically the structural elements of tau, red short pieces of alpha helix, and yellow short pieces of beta strand, but none of this is stable. So all of it is very floppy, mobile around. You have to imagine tau wobbling around all the time, not steady, unless, of course, it is bound to microtubules. We'll come to that. So, so it basically confirms on the NMR level that tau is a highly labile structure. The two yellow things here are the two hexapeptide motifs that are essential for the aggregation of tau. These are the only places in tau that have a halfway hydrophobic character, and that's and high beta propensity, and that's why they're also responsible for the aggregation of tau when aggregation happens with the help of some other cofactors. The slide also shows some of the many. So we're talking of the C-terminal domain as the microtubule assembly domain because it sticks to microtubules, and the projection domain is called projection domain because it projects away from the microtubules, and the function of the projection domain is really poorly understood, except that it contains a lot of interesting binding sites for other proteins like FIN, SARC, PI3 kinase, HA3 domains, PIN1, etc., signaling molecules in general. So this is a proline-rich region. You can see polyproline-like structural elements here, and so there's probably a signaling going on here. FIN, in particular, has been investigated by a number of people, but by and large, this area is still underdeveloped and worth pursuing further. The N-terminus is also interacting with some interesting cytoskeleton-associated protein, for example, dynactin, annexin A2, which is a membrane-associated protein, etc. So you can see there's a lot of interaction here in the microtubule binding domain. There's interactions with chaperones, with phosphatases, with other signaling molecules like calmodulin, 1433, RNA and lipid micelles. RNA very important because Tau may be a component of the stress granules, may be an important parameter of the stress granules, as recently published by Ben Bolosin's group. So there's different functions encoded in the different parts of the tau molecule. Just going quickly through some of the pictures, these are tau molecules in negative stain by electron microscopy. These are tau filaments by atomic force microscopy, more or less showing that it has there are filaments with a super twist which fall down. Now, just defining the assembly, the steps in the assembly process in global terms is you can think of a monomer which is unfolded. The monomer has to come together to a tau dimer. The tau dimer is actually can be stabilized by dimerization using the SH groups in tau. And then after that, there's a conformational transition which can be induced by, for example, polyanions. And then that forms the initial nucleus of beta strands called cross beta structure, because the filament is actually in a vertical direction here in this diagram, and the filaments run across the axis, and therefore this is what is called a cross beta structure, and that's what's similar to for many amyloid fibers. So this typical kind of cross beta structure in that regard is similar to A beta filaments and many others. Now, the implication of that is if you want to interfere with this toxic pathway of aggregation, then you can do two things. You can either knowing what the elements are that cause the aggregation, namely these hexapeptide motifs, you can either change those hexapeptide motifs so that they cannot aggregate, and one way of doing this, at least in vitro or in animal models, is to put in proline mutations, because the residue proline is not compatible with beta structure, or you can find small molecules that interfere with the self-assembly of tau, the PHF inhibitors or tau aggregation inhibitors, and this is an avenue that's pursued by a number of labs worldwide, trying as one approach to stop the pathology in Alzheimer's disease or others, and of course the methylene blue that was mentioned earlier on today, LMTX, is one example of this. Now, I just would like to remind you that there are several compounds that are useful in monitoring the aggregation of tau, and these compounds are named Congo red, thioflavin, and many others. They usually bind along the vertical axis of the filament, and the reason why they bind along the vertical axis is they squeeze in between the residues that are lined up as a cross beta structure, and when they squeeze in, that changes their fluorescence properties, and because of that you can observe the assembly of particle filaments or other amyloid fibers by the fluorescence properties, in the case of tau particularly by thioflavin S or T. Some more atomic force microscopy studies done in Daniel Müller's lab by Susanne Wegmann, showing that the tau fiber, when you look at it at higher resolution, it has a peculiar substructure of about 15 to 30 nanometer periodicity, not very well defined, but you can see it here. And when you now fragment the fiber, which you can do in two ways, one is you put the fragment on a hydrophobic surface, and in this case the parahelical filaments fall apart. They fall apart into these little peaks here, and the little peaks, if you look in between, there are little strings in between. So it's as if there are little packages of tau protein that are linked together by something very flexible. And you can achieve the same thing by simply running your AFM probe across the filament and break it apart, and again you see these fragments connected by thread-like elements. I'm saying this because the internal packing of the parahelical filaments, in spite of its apparently irregular structure, is still unknown, because again we've tried all sorts of things. Crystallization didn't work, NMR doesn't give you results, solid state NMR remains ambiguous, so we're still at a point where we know generally there's a cross-beta structure, but how the elements of tau protein are distributed within a filament, we don't really know, and therefore it's difficult to predict what kind of approach you should take in order to break apart the filaments if you're looking for tau aggregation inhibitors. This just shows an assembly study of regular tau with time. When you let it incubate, in 48 hours you get nice filaments. And if you have a mutation that promotes tau assembly, you get actually many more nuclei before you get filaments. This is the delta K280 mutation. This is interesting because a lot of people think, or the general consensus is that it is not the tau filaments that is actually toxic to the cell, but it's the things that happen on the way, the oligomers. Broadly speaking of oligomers is a way of saying I don't know, but they're small and they can usually not be spun down by ultracentrifugation, so it's probably a broad spectrum of things, but you can see the image of that by atomic force microscopy. And again, by atomic force microscopy, if you take the tip and let it down on a tau filament and ask what is the resistance of a tau filament against this tip, you can, first of all, you get this picture here. You have a core, and it's surrounded by a floppy coat of things. And the scientific name of a floppy coat of things is soft polymer brush, meaning it looks like a brush, and but it doesn't offer any resistance. And the brush actually comes in two parts. First of all, you have a fairly rigid core, and you have a more or less a basic inner part of this surrounding, and then a more acidic outer part of the surrounding. The more acidic part on the outside comes from the fact that the C-terminal part of the C-terminal 120 residues of tau are predominantly acidic, whereas the rest of the molecule is basic. So that's roughly the disposition of the elements in such a fiber. Now let me talk about the regular motives for controlling aggregation. I mentioned before the elements, the hexapeptide motives that are responsible for the aggregation. These are elements at the beginning of repeat R2 and repeat R3. I should say there are two nomenclatures. If you read the tau literature, you will find that people use two different definitions of what they call a repeat. If you have 4 repeats, of course, in principle, it's pretty arbitrary, but one way which we follow is actually the terminology whereby repeat 2 corresponds exactly to exon 10, and then that aligns all the other repeats as well. So at the beginning of repeat 2 and beginning of repeat 3, you have these hexapeptide motifs, VQINK or VQIVYK, and if you mess around, they have an enhanced potential for beta structure, and if you change these peptides, then of course you affect aggregation. And Martin von Bergen, who discovered this in the lab, he coined the term pro-aggregant mutations and anti-aggregant mutation. The pro-aggregant corresponds to the mutation delta K280, which is observed in tauopathies, which enhances the beta propensity of this element. And the anti-aggregant, simply you stick a proline in here, you stick a proline in there, and you get no more aggregation. And the reason why this turned out to be important in our experience is that transgenic mice or transgenic worms or transgenic anything, if they have pro-aggregant mutations, they get sick. And if you just do the anti-aggregant, they stay healthy. So that is a very simple way of manipulating the state of disease. If tau cannot aggregate, and in fact this protein does not aggregate in these different animal models, then you have no effect of tau. You can overexpress it. There may be slight effects of overexpression, but the animals don't get sick. So therefore, this is a rationale for looking for tau aggregation inhibitors, which what we and others are doing. So now coming back to how tau interacts with microtubules, I think there is some interesting story to be learned here. This is again results from the Zweckstätter Griesinger lab, Karavat et al., who have managed a way to get at the structure of tau elements bound to the microtubules. Don't ask, I will not go into the details how they did it. I'm just showing you the tubulin structure as solved by Gigon and Knossoff a few years ago. So we know the tubulin structure in a crystalline form. We can draw models. We know where known microtubule binding molecules are located. For example, this is the so-called vinblastin site. This is the colchicine site. This is the Taxol site. And it turns out that if you probe, if you ask which parts of tau bind to these sites, then it turns out that an important part of tau binds to the vinblastin site. The vinblastin site happens to be the site right in between the two alpha-beta heterodimers of tau. So this is of tubulin. This is one alpha-beta heterodimer. This is another alpha-beta heterodimer. The two, you stick to the two together, that's how a microtubule assembles, and right in between, that's where part of the tau binds. And I show you an example of what the tau structure is when it binds in there. This is the peptide that contains the beginning of repeat R2. This is the peptide showing the beginning of repeat R3. They're both similar in structure, and this is a more artistic view of it. So what you see here is the PGGG loop before the hexapeptide motif that is responsible for aggregation in a beta structure here. So loop and beta structure shown here in an extended form. It becomes a real beta structure only when it interacts with another tau molecule. So that's very important because it explains, let me see, I want to get the next one here. It explains the properties of an antibody that I think is highly interesting. Yeah, here. So Michal Novak in Bratislava, the name was mentioned already today. They're in phase 1, 2. They're in phase 2 with their active vaccine, and they have an antibody called, they looked for antibodies that interfered with the microtubule assembly. They found one they called DC8E8, Konsekova et al. And it turns out that this antibody actually binds at the part just before the hexapeptide motifs that cause aggregation. So if you just look at this loop and extended structure here, the antibody epitope would be roughly here, and this would be the part that actually then causes aggregation. So the antibody by fixing the loop prevents the aggregation of tau. So it's a perfect aggregation inhibitor, and therefore it makes sense to develop a vaccine against it, which works surprisingly well in mice and rats so far, and of course in vitro as well. So I think this is a good example of a link between an immunological approach to find something that works against tau aggregation and the structural equivalent of it. So we'll see, they actually haven't revealed the structure of the antibody, so but we can guess, we can have a pretty good guess that it is actually here where the shadow is. So, it makes rational sense to look at antibodies, look at where they bind, and find a structural reason of why they work the way they do. Now let me say a few words about transgenic animals. This is mostly the work of Eva's group, so I'll just go quickly and leave out some slides that I don't understand. So here is, again, the tau structure with the hexapeptide motifs. This is where the delta K280 mutation is, and most of the models we made consist of placing pro-aggregant tau into a mouse or an anti-aggregant tau, and then comparing the two. And this is done in a inducible way, controlled by doxycycline, basically, so we can switch the expression of tau on, and then we can observe the aggregation of tau and the development of pathology, and we can also switch it off, which is a nice part of this application. So you switch the tau off, the tau disappears, and you can ask the question, does the disease go away? It does, actually. So now, first of all, showing you a pro-aggregant mouse, 3 months old, has a lot of tau deposits in the brain. An anti-aggregant mouse, even though it's 22 months old and very aged, shows absolutely no aggregation, which shows you the power of replacing a single proline in an aggregation-prone filament. You get rid of aggregations. It's perfect cure on the on the level of molecular biology, at least. So the properties of these regulatable pro-aggregant tau, and I forgot to say the other nice thing about these mice is that they co-express luciferase, so you can actually look by bioluminescence at what stage the animals are. So you see a fluorescent, a bioluminescent mouse, and you know it's going to be sick a year from now. So you can actually test treatments on these mice. Anyway, these mice show hyperphosphorylation, all the usual things you expect of Alzheimer tau. The most important thing is they show synaptic decay, loss of LTP, cognitive impairment, and when you switch off, and that's the main thing here, when you switch off this tau, in this case we do the tau repeat domain, but we also do the full-length tau, then you get synapses back and you get cognition back, so you can cure the memory deficit that these mice develop. And by contrast, the anti-aggregant tau mice don't really show anything. They just don't care about the tau, as long as it doesn't aggregate. This is an example of a water maze experiment. A regular mouse finds the platform in 10 seconds. The demented mouse, after, I don't know, this is now 12 months switched on, it goes around and round in the basin, but can't really find the platform, so it really has lost spatial memory. But if you switch off tau for just a few weeks, the memory returns. And so therefore, it means that if you get rid, or if you find a way to neutralize this toxic tau, you're in business as far as treatment is concerned, at least on the level of mice. The other interesting thing is, of course, that this tau that aggregates not only by itself, but it also takes the mouse tau and causes co-aggregation of the mouse tau. So what you wind up with is a toxic mix of mouse tau and human tau, in this case repeat domain, and this is the thing that is toxic to the mouse. Whereas if you switch off, then by dynamic instability, these filaments gradually exchange subunits. But since you do not express the toxic tau anymore, gradually you wind up with a pure aggregate of just mouse tau. But the mouse tau, even though it's still aggregated for quite some time after switching off, is still allows the recovery of memory. So the mouse tau, even though it's aggregated, is no longer toxic. The explanation of this phenomenon is probably because the oligomers basically disappear. The fibers stay around, but the oligomers disappear, and they are the toxic elements. So, and the other thing that, of course, is the important observation is that synapses, initially when you express this pro-aggregant tau, synapses disappear, but when you switch off, then the synapses reappear, and that nicely explains the recovery of memory. So implications: learning and memory can be restored if one neutralizes the misfolded toxic tau, and this provides a justification of using compounds or antibodies to inhibit aggregation. I show you some experiments with this type of transgenic mouse where we try to test methylene blue, which is sort of the parent molecule of the LMTX checked by Claude Wischig and colleagues. Actually, I heard about this thing for the first time at the meeting in Barcelona many years ago. We had a FEPS meeting, I believe it was, where he for the first time talked about this thing. I think you organized a session, right, or something. Anyway, so the kinds of experiments we do is we know that the mouse left alone would get cognitively demented at age, let's say, 12 months. So we go in with methylene blue, give it in the water, and ask how much earlier do we have to give methylene? Can we actually prevent the onset of cognitive decline, and how early do we have to go in? And the answer is, even if you go in only relatively shortly before, so the blue arrow shows different time points going in before, even only shortly before, let's say 2 or 3 months before, you can totally prevent the development of cognitive decline. However, if you do this after the decline has started, you can no longer prevent it. And the explanation for this is this somewhat complicated diagram. imagine that tau, when you express it, aggregates and aggregates and aggregates, and at some point, cognitive decline will begin at the this is the threshold, basically. When you switch off, in the case where we switched off, you can actually reverse the production of tau and actually get below the threshold. So that's the blue line here. But if you just put in an aggregation inhibitor, it decreases the rate of increase, but it doesn't reverse it. And therefore, but if you decrease the rate, this rate sufficiently, then the threshold of cognitive decline will be reached only after the death of the animal. So the animal will never experience cognitive decline. That's why giving methylene blue, even relatively shortly before, has the effect of protecting the mouse against cognitive decline. So I think that's basically what we would like to also have for a drug. If we could have a drug that makes me sort of demented at age 120, I wouldn't worry too much about it because I'm going to die of other reasons anyway. So, so it shows it's possible. So now, just as a way of comparison, we also looked at the A152T mutation, which was a mutation that turned up with a patient who's actually the founder of the Tau Consortium, it also turned out that this is a risk factor for PSP, and therefore, so you can ask, what does it do? Because it is outside the repeat domain, and therefore the question was, what are the causes of this? So we developed a mouse expressing tau A152T in parallel with Leonard Booker's lab. We did a slightly different mouse, and the experiments actually agree quite nicely. The one thing is that this A152T aggregates, but not as well as normal tau, and the aggregates are much more brittle. I won't go into this, but less stable anyway. If you overexpress, or not overexpress, but if you express this protein at low levels, actually, you still get tangles in the mouse, but at a much later stage, you also get hyperphosphorylation. But the main thing you get is you get pronounced astrocytosis and microgliosis, and you get excitotoxicity and epileptiform activity in this mouse. So here, an example of astrocytosis shown with the IBA1 antibody here and the dentate gyrus part, a very striking phenomenon. And so what we did is, what Eva's group did, she coupled this effect with the expression of luciferase under the GFAP promoter, so she could see when the inflammatory response would start. It turns out that the inflammatory response starts at about 5 months, whereas cognitive decline occurs about 15 months. So we know 10 months before it occurs which mouse is going to be suffering from pathology. And this, of course, the early detection enables the monitoring of the efficacy of therapeutic treatments, which is what's going on in the lab at the moment. But the other remarkable thing about, well, so let me first say this. These two mice that I just described have totally opposite effects as far as neuronal activity is concerned. Neuronal activity measured here by calcium increase after chemically induced depolarization. And in the case of the delta K280 mouse, calcium essentially decreased. There's loss of LTP, and the neurons show a reduced activity, whereas in the A152T mouse, we get the opposite, a very strong response with the A152T and corresponding hyperexcitability. And the explanation, I'm leaving out a few slides. The explanation is basically you have to imagine that this is the presynaptic part of the synapse. Tau is somewhere in the axon. It's still primarily in the axon. However, the effect of it is that when there's excess glutamate released here. The excess glutamate sort of is normally taken up First of all, it causes calcium increase, and calcium, depending on how much of it is, can have deleterious effects. For the deleterious effects, in particular in this case, the activation of the extrasynaptic NMDA receptors can cause excitotoxicity, Krebs activation, and cell death. Normally, the glutamate is taken up again by the astrocytes with the glutamate transporters, and you can actually cure the effect, the hyperexcitability. You can cure the effect by ceftriaxone, which is a way to increase the efficiency of the glutamate transporters. And you can, of course, also treat these synapses basically with memetin and ifenprodil, which are both drugs that work on the extrasynaptic NMDA receptors. So, I think I should sort of stop here. We have the same thing on the level of C. elegans. C. elegans moves nicely around, but when it expresses a tau in the neurons, and which gets aggregated, then it gets paralyzed. And we use the C. elegans as a cheap way of monitoring drug effects. I can't go on here. So I won't go into this, but just show you when they're paralyzed. This is where the mice have the C. elegans, the worms have aggregated tau in their neurons. But when you treat them with aggregation inhibitors, then they move quite nicely around. So this is an easy experiment to do, because the worms take up the drugs much better than, of course, the mice, because there's no blood-brain barrier. So you can check whether an aggregation inhibitor actually works. You can check this relatively quickly, and this works well for the delta K280 mutation. But I'm just showing you the other effect with the A152T mutation. We can tell that this toxicity is very different from the toxicity that's exerted by the aggregation, because here we also get paralysis, but the paralysis is not based on aggregation anymore. You don't have aggregates, and you cannot treat by aggregation inhibitors. So we find this mouse particularly interesting, because it leads us to think that there may be some signaling mechanisms that are going awry here in this worm, and this is worth investigating, because of course there are many interactions that tau can have. So I think I was going to. I think I'll stop here. I have some slides here on spreading of tau, but let me just go through it. There's so many different ways of spreading, different mechanisms. If there's interest, we can talk about that in a way. It's a totally bewildering business at the moment, so let me just skip through it and come to the very last slide I have to show. So this is the team at the German Center for Neurodegenerative Diseases, and also at the Institute CSAR, is currently still located in Bonn. Here, the Institute DZNE is moving to a new building, or is in the process of moving to a new building at the Medical School of Bonn University. And our Queen equivalent, called Joachim Gauck, was just, he's the president of Germany. He just visited and said some nice words and said we should find a drug before he gets really old. But he's not going to run for another presidency, which is a pity, probably because he's aware of his age. So thank you very much for your attention.
Thanks, Eckhard. So we have time for one or two quick questions, very quick ones.
My name is Justo Yevenes. Thank you very much for your presentation. It's very nice. We have shown a number of years ago that in the absence of parking, tau distribution changed from the axon to the perinuclear region. My question is if there is any evidence that under certain circumstances, tau could be a transcription factor or change transcription in any way.
So one part of the answer is that yes, the observation that tau can change distribution is a very important observation already made by the Brack in his the Heike and Eva Brack in the early 90s, which is a very early indicator of the fact that something is going wrong. Now I think it can have different reasons. Tau is a very sensitive indicator of stress, of anything that can go wrong. It can have probably different explanations. One explanation is that tau can move into the axon, but it can also move out of the axon. That has to do with the barrier of tau at the axon initial segment. The other explanation that we currently favor at the moment is that if tau gets missorted, the term that is often used in this context is missorting. When tau is missorted into the cell body and into the dendrites, that has to do with the fact that the axonal transport does not work properly anymore. So it's synthesized in the cell body, but it doesn't go away into the axon and therefore accumulates in the cell body. This is, for example, the case if you challenge neurons in cell culture with A beta, for example. We initially thought missorting comes from the idea that you have something that's sorted into the axon and now it's missorted into the somatodendritic compartment, but actually it never gets into the axon, it just stays in the cell body, and that may very well be because of something that has to do with protein synthesis or could have something to do with stress granules, with cells under stress, where then the proper sorting of your molecule to the tiny hole that is represented by the axon cannot be done anymore. There's a very nice paper by Hirokawa's group, I think Nakata Hirokawa, about 2002, who describes how difficult it is for a soluble protein like tau to actually find the axon. I mean, you're on the surface of the Earth and you want to climb the Eiffel Tower. How do you find the base of the Eiffel Tower? The fact is, normally you don't find it. So there's an intricate mechanism that needs to guide axonal proteins into the axon. And if something is messed up there, then it's easy to see why it gets missorted. There's a long answer for saying, I don't really know.
Eckhard, proaggregant tau in the presence of heparin results in the appearance of many small aggregates and some few filaments or large aggregates. The question is, if you add methylene blue, is faster the destruction or the inhibition for the small aggregates or for the larger filaments? Have you studied that?
I think we never addressed the question the way you do. I mean, we looked basically always for thioflavin fluorescence and looked at the increase or decrease of thioflavin. It's certainly an interesting question. I would imagine that the small aggregates disappear faster, but I don't really know. What Eva knows better.
We looked at it. I mean,
okay.
We threw, of course, methylene blue onto fibers and oligomers, and then they partly dissolve. Yes, in EM and in AFM we did this.
Yeah, but the question is which one dissolves faster? And
okay. Sorry.
I think actually, it would probably be very difficult because, which technique do you use to actually make that statement? AFM presumably, or
Like you saw us the thing that happens when you put heparin and without methylene blue. In the case of the wild type, you have fibers. In this case, you have a lot of the small ones and a few fibers. If you do with wild type and with the proaggregant, perhaps you have a relative easy answer. Because I think that could be important because it's about the importance of the, as you said, the oligomers, or it's the importance of the filaments. That probably everybody said it's more important the oligomers than But it's not really demonstrated.
Yeah, I mean, and of course it gets even more complicated. There's a recent paper by Sigurdsson's group who has developed an antibody that binds only to the monomer, and that's the only antibody that works in his. Although there are some issues with the paper, I think it would need to be, we can discuss later, but anyway, it's really very complicated field. Yeah.
Okay. All right, thank you very much.